family and award committee for bestowing this high honor on me. I have 400 colleagues that I speak on behalf of at both the Venture Institute and Synthetic Genomics, and I accept this award on behalf of all of my colleagues. I'm going to give, hopefully, an overview of the work going on and that has gone on at the Venture Institute and also at Synthetic Genomics, starting in any time now. Here we go. Well, it's almost there. So the Venture Institute is located both in Rockville, Maryland, and in La Jolla, California, and also Synthetic Genomics is also located in La Jolla. And also we are now building what we understand is the world's first carbon neutral building on the University of California, San Diego campus as part of the Institute to try and put our ideas of sustainability into practice at several levels. So this building, which will be completed in October of next year at UCSD, will generate all its own power and recycle all its own water. We haven't got approval yet from the city of San Diego to let the drinking water come from human waste, but we invite you to come have a drink there sometime in the not too distant future. I think what all of us have been doing in genomics, and particularly what I want to talk about, is conversion of biology into the digital world. So I view DNA as an analog molecule and an analog information molecule. And when we sequence that DNA and put it in the computer, we're converting it into the ones and zeros of the computer. And that's been a challenge now for several decades. I think really starting with Sanger's work in 1977 of sequencing the Phi-X genome. And as we've been accumulating more and more of this data, we have a new challenge to, in fact, try and prove that this information we've been generating actually represents what we think it has. So we've started trying to go in the other direction, starting with the binary code in the computer and seeing if we could then remake the analog molecules and regenerate life out of that. As you heard, this got underway in earnest on the digitization of biology, starting with new mathematical approaches based on EST sequencing, where in 1995, Ham-Smith and I and colleagues sequenced the Haemophilus influenza genome, and this was doing a shotgun sequencing with a mathematical reconstitution in the computer. 5 years later, we were able to scale up orders of magnitude and do the same thing with hard draft of the human genome. Now, this is the team that we had at the White House in 2000, along with Francis Collins and President Clinton and Tony Blair. The tall man with the white hair is Ham-Smith, who discovered restriction endonucleases and got the Nobel Prize in 1978. The dark-haired guy between him and me is Gene Myers, who led the team for developing the new algorithms and writing over 500,000 lines of computer code in less than 6 months for the new algorithms, basically the basis of what everybody has used since. The Macaulay Culkin-looking guy is Mark Adams, who led the team for automating all the approaches. This is the type of lab we all used to have. With hundreds and hundreds of machines. At Celera, we had 300 of these. This was the post-Celera lab at the Venture Institute. It was massively parallel automation. On the computing side, in 1999, we had to build the third largest civilian computer, which was 1.5 teraflops. Obviously, the world has changed a lot since then. We just had the 10th anniversary celebration, and this slide proves that people look better in tuxedos. But that giant room, the giant rooms that we've all had, have now been condensed to things smaller than this podium. So this is the new ion proton sequencer out of Life Technologies that can do a genome in about 2 hours, making it useful for clinical work. Everybody talks about the cost of sequencing. So this is $1,000 to $2,000, but we still need about $10,000 worth of bioinformatics work to analyze the data. But it helps because that giant room with 1.5 teraflops is now reduced to a simple board to add to your PC at a teraflop each. In 2007, we expanded it to do the first complete diploid genome. In 2000, everybody just did half the genome because Lee Hood estimated it would be $1 a base to do the genome. Jim Watson decided asking Congress for $6 billion was too much. So we just do half and assume that the other half was reasonable, would be very much the same. But it turned out, in fact, to be different. Instead of one base out of 1,000 being different, we're now looking at the whole genome, my 2 parental chromosomes different by 0.5% from each other. So if we're comparing any 2 individuals, we're actually comparing 4 sets of chromosomes. So we're all in the 1 to 3% range. Vanessa Hayes at the Venter Institute has been studying human evolution and diversity. She sequenced the Bushman genome out of Africa and Desmond Tutu's genome, and is actually tracing back humanity to the oldest origins in Africa using this data. And when we compare my genome to a Bushman's genome to a Han Chinese, we find we're all in this reasonable Venn diagram of expectation. When we look just within Africa, it's what people have known for a long time, that there's a lot more diversity within Africa than between Africans and the rest of the population. It's in part because some of the groups that migrated out of Africa that led to all of us, some of those groups migrated back and intermingled, and it turns out Svante Pääbo's data showed that a few of those stopped to have sex with Neanderthals along the way, making the populations even more complicated. Those were my ancestors because, in fact, the team that sorted out the Neanderthal genome proudly used my genome to show I was roughly 3% Neanderthal. President Clinton quotes that all the time with his own genome, and it said it explains all his deficiencies. So I will take the same excuse. But just looking at half a genome is not very useful for understanding heredity. Understanding that the complete diploid genome is. And we've been taking a number of approaches to this as others, but just to show you how the technology is advancing, Roger Laskin at the Institute developed what people are now doing with single-cell genomics. We can actually sort single sperm cells and do a genome on a single sperm cell. Sperm being haploid, we've shown that there's about 1 crossover per chromosome. So I keep using the bad analogy. I say if you do a handful of sperm cells, you can actually get easily sorted out the chromosomes in a haploid fashion from parents. And compound mutations actually going to be far more important than many people thought early on in explaining genetic traits. I have the pleasure with my genome being out on the web, but people are constantly publishing data of how many genetic disorders I have or I'm likely to have. And just looking at compound mutations, there's a lot more that shows up as significance instead of just looking at single gene mutations. Some of these are starting to have an impact on medicine. The pharmaceutical industry has been the slowest to adopt genomics and genetics because they thought it would lower their sales. They were right, it has. And an example is Pfizer's compound to try and treat lung cancer failed its clinical trials. There wasn't statistical significance until a team of scientists found there was a single mutation in a gene that if you gave the same drug to cancer patients with that single mutation, over 60% of them had significant tumor regression. Pfizer made up for it by charging $10,000 a month for the drug. So the pharmaceutical companies adapting very quickly to genomics by still getting blockbusters, but by going up exponentially in what they charge for pharmaceuticals. So it's not clear how that model will work in the long run. We found we're not alone with our 100 trillion cells and our 20,000 some odd genes. When my team, led by Karen Nelson, first did shotgun sequencing in the human gut, it led to this new field of the microbiome. And it turns out we have around 200 trillion microbial cells and maybe 10 million additional genes associated with each of our bodies. So it doesn't matter how often or how completely you wash your hands, you're not going to get rid of all these microbes. So the question is, what roles do these play? Do they play a role in human disease and wellness? Now NIH has started funding this on a massive scale, characterizing with the new sequencing all the microbes as references. And with new multiplex sequencing, the very first genome we did cost us about $1 million to do. We can now multiplex microbial genomes and do full genomes for roughly $58 a genome. So it's getting more economical to do these studies. But changes in the microbiome are starting to show up with more and more diseases and conditions. My favorite effect that was discovered is most people think alcohol damages the liver by effects of alcohol in the liver. It turns out the main damage is due to alcohol effect on a bacteria that releases a toxin that damages the liver. So being a non-physician, I've come up with the notion that we should fortify alcohol with antibiotics, and therefore we won't have any liver damage. But that hasn't caught on broadly yet. So the other question is, aside from a disease association, what is the metabolic potential of these tens of millions of additional genes? And the new field of metabolomics is starting to characterize this in detail. And Metabolon in North Carolina has been characterizing references for human chemicals and sorted out there's roughly 2,500 chemical compounds that we as a species can make in our metabolism. So if you look in your bloodstream after a meal, you find about 500 or so chemicals circulating in the blood, a lot more than people thought. But only 60% of these are from our own human metabolism. 30% are from all the chemicals from your diverse diet. But about 10% or 50 chemicals circulating through your brain right now are for bacterial metabolites, both of the chemicals from your diet and from your own human metabolites. So the saying is, you are what you eat, but we're not only what we eat, but we are what we feed our bacteria and what they tell us. So how do we go forward from here? It's been very limiting what's happened in genetics, looking at single genes, looking at focused disease studies. They can tell us whether there's an increased risk associated with certain changes in genes, but it doesn't tell you why you don't get those diseases if you have those same changes. So obviously we need to look at the entire genome, but we need to get a digitized human phenotype and do this tens of thousands to tens of millions of times to get a database that's significant enough to start making statistical references for individuals. And so I think that's the challenge as sequencing has come down exponentially. Trying to put together human knowledge in a way we can link it to sequence is a challenge that may be far tougher than the human genome. You heard in the introduction that we, aside from the human gut, we did a shotgun sequence into the oceans, and many of my colleagues, particularly those in the Department of Energy doing the same study on acid mines, think my greatest discovery was finding a way to sail around the world on my own boat and do science. And so we took samples every 200 miles, filtered all the microbes out of the water, and just shotgun sequenced those. and started to characterize those. And at the time we started this, there was a paper published in PNAS saying there was very little microbial diversity in the oceans, and there was much more in soil, but the oceans were very uninteresting. In fact, the microbes discovered in the oceans were numbered in the single digits. In fact, we started in the Sargasso Sea because that's where a lot of these were first discovered. SAR-11 was the 11th microbial species discovered there. And we stopped sequencing after we found about 1.3 million genes that hadn't been seen before. And at the time, there were less than 1 million genes known in the databases. And that led to the expansion of taking samples around the world. First comprehensive results were published in this special issue of PLOS Biology a few years ago. We spent the last couple of years sampling the North Atlantic and the Baltic and Mediterranean seas and up into the Black Sea. So these were positions that were well sought after, having to sail from Italy into Greece and up through the Bosphorus into the Black Sea. But what we found by looking at the data around the globe, that there was much more variation in the genomes of the microbes than anybody imagined. SAR-11 was thought to be a single species. But from shotgun sequencing, and this is a simple graphical tool where we have a reference genome across the top and compare each of these tiny bars as roughly 600 base pairs of sequence to that reference. And you can see instead of the normal band of 1 to 3% variation you might expect, we get up to 40 or even 50% variation. Because we have the nearest neighbor, a typical bacterial gene is 900 base pairs. On the average, we get 1.4 genes per sequence. And so we know the nearest neighbor. And these genomes basically have the same genes in the same order, but not with 10% variation like we have for all mammals, but this tremendous variation that you see here. In fact, as we add more and more data, what people thought were single organisms turn out to be entire taxa. The third largest taxa thus far known was never cultured before. It's SAR-86. It was seen by a 16S RNA signature, but we've now been able, from all this data before we're able to get his genome and culture it, to just assemble it from what we call an extreme assembly from all this data around the globe. We've been doing the same thing in the air, only it's been substantially more difficult because we have to deal with very large volumes of air. Imagine filtering all the air in this room and isolating the microbes and viruses from it. And we found it was very easy to get contamination with these machines, but this is similar to what we did in the oceans, where it's a cyclone vacuum cleaner basically that puts things in solution that we can then filter through these filters and do shotgun sequencing. There were inhibitions to this. We found in the air in New York City, we couldn't get DNA sequence until we found that the samples contained a huge amount of iron. And we simply used a magnet to pull out the iron, and then we could start to get good DNA sequencing. So you can imagine that there should be very low anemia rates in New York City. It's not clear where all the iron comes from. It's assumed from the construction and all the train rails. But we were sampling at the level of the 22nd floor, inside and outside this building in Midtown. And we also sampled in San Diego in a hospital and also in a home and off the end of Scripps Pier. And we've now worked out a deal with NASA where they're giving us all the HEPA filters over time from the space station. So you imagine if each individual going up is taking 200 trillion microbes with them from the different environments of people going to the space station that can get rather ripe up there. And it turns out it's a real problem because each new astronaut or cosmonaut is bringing a whole new microbial population in with them. What we found surprised almost everybody because we're actually collecting things at the level of whole cells, not free DNA. So in New York City, inside this building, obviously human DNA was the majority of what we found because we all shed tremendous number of cells on a continuous basis. But you can also see we found about 15% of rodent DNA. This was a major financial firm, so that wasn't necessarily a surprise to a lot of people. You can see a lot of bacteria, plants, other animals, fungi. When you go outside at the 22nd floor level in New York, 46% of the cells in the DNA was rodent DNA. It turns out this is primarily mouse, not rat DNA. And as you go lower and closer to the street, the percentage of rat DNA goes up substantially. You can see San Diego is a much cleaner environment. That's why people like to live there. And the house and the hospital look roughly similar, of a lot of different microbes. You go outdoor in the hospital, you see plant DNA. And if you go to the end of Scripps Pier, depending on which way the wind's blowing, you get a lot of insects. We get marine algae and other things. The big difference comes when you check out what kind of organisms are in the hospital versus the home. We see a huge collection of virulent organisms in the air in the hospital. So it's no wonder you get sicker or can get sicker when you go to the hospital. You're exposed to all these infectious agents at a level that you would not be exposed to anyplace else. So this new way of environmental characterization is a way of understanding a lot more about our environment. During the course of this symposium, all of you are going to breathe in and out roughly 10 to 100,000 microbes, depending whether the person next to you is sneezing or not, or coughing, and at least 10 times that many viruses. The virus level that we found in the hospital was huge. Because we're limited on our viral databases, the main matches they hit in the databases were all the viruses that we got out of the human intestine. And so we don't know if that's biologically significant or just a matter of having limited viral databases at this time. If we look at rates of gene discovery, mammalian genes have basically been discovered. That you may find one or two additional ones. If you find a rare mammal, you might find a couple, but basically we've saturated the mammalian tree. If we look at the rest of biology, whether it's plants, insects, microbes, viruses, we're still very much in this linear phase of discovery. And so it's easy now for a student to go outside and just take a sample and make thousands of discoveries that weren't possible before. Actually, I was told at the start of my PhD work in the early 70s in La Jolla that it was going to be very hard to find something new in biology because it was essentially all known. Now we know how easy it is to make lots of discoveries, and as David pointed out, sorting out that information is a huge challenge. We're not just finding new members of known families, we're constantly coming up with new gene and protein families that haven't ever been seen before. So our diversity is growing, and the problem is we don't know the function of most of these. Structural chemists are trying to see whether these represent new types of protein folds or if that part of our domain has been saturated. And more and more of these new families we've discovered are being crystallized and characterized. There's a new field developing of stem cell genomics. Obviously, we can make iPS stem cells from skin fibroblast. But one of the things that's developing with all stem cells, whether they're embryonic or iPS cells, once you get the cells out of the human body, they start to deregulate. First methylation changes, then we start to get a lot of changes in the genomes. So it turns out context is important, and people are just starting to understand this, and people have made recent advances by, instead of using mouse feeder layer cells to grow stem cells on, of using immobilized sugars or other chemicals to find ways to stabilize the genome. But this is very critical and could be the Achilles heel of stem cell therapy if we can't solve it. And if we can't stop the changes, the question is, do they happen continuously? And people are studying neuronal cells made from my skin cells, and they have a number of changes. Some of them may be beneficial, so I'm looking forward to that. But we can actually use our new synthetic DNA tools to go and do gene therapy on isolated stem cells. And what stem cell scientists have been using for information, the kind of slides that David was showing his early history, where you look at the karyotype, that's the level of information that's been used. If it looks like a normal karyotype, it's assumed the cells are normal. the field could suffer the same fate as gene therapy if individuals get cancer from a stem cell therapy. Again, we can do high throughput automated single stem cells, including looking at transcriptomics on single stem cells and trying to understand these processes. And I've argued from the beginning, understanding stem cells is going to be key to understanding how our genomes actually function. And understanding our genome is going to be important for understanding stem cells. In 1995, we actually sequenced 2 genomes, that of Haemophilus, and the second one was of Mycoplasma genitalium, because once we had the first genome in history, we wanted something to compare it to, and we thought to make sense of it, having a simpler genome might help make sense. And this was around the time there was a lot of discussion of what minimal life might be. In 1996 is when the NASA team announced they had nanofossils in a Martian meteor, and so questions were around what would be a minimal life form. A simple calculation showed that those were too small to even house a single DNA or RNA molecule, but we started asking new questions. How many of these genes are essential? Doing the first comparative genomics. What's the smallest number of genes to run the cellular machinery as a means of understanding all these functions? And after several years of trying a variety of knockout things, because mycoplasma don't have all the lovely tools that yeast have, we decided the only way to really approach this was to design and construct a minimal genome in cell. And as soon as we started down that route, we had new questions. Would the chemistry even allow us to make these large DNA molecules? And if they did, would it just be a large DNA molecule? Could we find some way to boot up the software? So after the human genome, we went back to this problem in a big way, and Clyde Hutchinson had been a postdoc in Fred Sanger's lab and took part in sequencing Phi-X 174, and so we decided historically to start back with that, and also because Phi-X can take very few mutations in its genome. The problem with DNA synthesis, those of you who do it, is an error-prone procedure. It's n-1, so the longer you make the oligonucleotide, the more errors there are. So we set out new ways to do error correction. We started with the digital code of the genome in the computer, 4 bottles of chemicals, and we made the 5,000 some odd base pair of Phi-X genome. We then took that DNA, injected it into E. coli, and what happened is the E. coli machinery recognized this DNA immediately, started making Phi-X proteins. They self-assembled to form the virus. The virus showed its gratitude by killing the cells that made it, which is how we detect it with these clear spots on a bacterial lung. So we call this a situation where the software is building its own hardware. We just put in this chemical software piece, and it got converted first into proteins and self-assembled into the virus. So we knew we could make 5,000 base pair pieces accurately. We wanted to make a 600,000 base pair genome, so we thought if we made serial pieces accurately of Phi-X size or viral size, we could find a way to put them together to make the entire chromosome. So we worked on a number of assembly methods, and my key colleagues in this are Ham Smith and Clyde Hutchinson, and then a postdoc, Dan Gibson, joined, but we have a team of about 25 people. So this looks like a basketball playoff where we made these 6,000 base pair pieces. We sequenced them, made sure they were accurate. We put 4 of them together to make 24,000 base pair pieces. We cloned these in E. coli, isolated, sequenced it, made sure each step was stable, and went on to the next stage. And this was extremely slow, as you can imagine, with each step. And we got up to 144,000 base pairs, and all of a sudden E. coli wasn't so happy with these really large synthetic pieces. We could actually clone the quarter molecules in E. coli, but when we tried to go up to the half genome size, we really started to have a lot of trouble. So we looked around and at a number of other species. I should have just called David because he would have told us we should have just gone to yeast immediately. And so the hero of the story is actually Saccharomyces cerevisiae, and we're able to use its homologous recombination tools to assemble the genome. So we simply put in the quarter molecules, synthetic molecules. with a simple yeast vector containing a synthetic yeast centromere. And it turns out the major difference, at least for growing these bacterial chromosomes in yeast, is adding this yeast centromere. And maybe that's going to be one of the central differences between prokaryotic and eukaryotic genomes, but I'll show you more about that in a minute. Putting these pieces together, yeast automatically assembled them using the homologous recombination. And on sequencing it, we found that we had the complete Mycoplasma genitalium genome as designed and built with this system, and that's what we reported in 2008. We continued to improve this synthesis technology because this was so slow. It was like the early days of sequencing. If this is all we could do, this was not going to be a very viable field. Dan Gibson made a tremendous breakthrough by simply putting the three enzymes we were using together and selecting the source of those enzymes carefully in a single tube at 50 degrees centigrade, which allows us now to be a single-step reaction. All you do is put in the designed oligos, incubate for a few minutes at 50 degrees, and you get assembly. And this obviously allows automation because it's now a single-step reaction. So we can go from the digital code in the computer to making analog molecules in a completely automated fashion without human intervention. We tested this early on just by synthesizing the mouse mitochondrial genome. So you can now buy this, we named it after Dan Gibson, the Gibson Master Assembly mix, and students around the world that are taking part in the iGEM competition are the biggest consumers of this. You can buy this now from New England Biolabs. So a much more complicated question. We were solving the chemistry. We had two teams, one concentrating on chemistry and the other on the biology. So how do you boot up a synthetic chromosome? It was pretty easy in the case of Phi-X or a virus where the DNA is infective. When you don't have infective DNA, it's much more complicated. And also these large DNA molecules are very fragile. You can't pipette them. We have to move everything around in gel blocks. We do the enzymatic reactions in gel blocks, and we have to move them in and out of gel blocks to get them into a cell. But this paper we published in 2007, I think, is one of the most important that we've ever come up with because it tells us really fundamental things about biology that apparently a lot of people didn't know, although Turing and others predicted things like this a long time ago. Simply by changing the genome in a cell, we converted one species into another. And because this is core to synthetic genomics, let me walk you through what we did quickly. So we had two related species of mycoides, of mycoplasma, M. mycoides and M. capricolum. They're roughly the same distance apart that we are from mice. When we were first doing this, we didn't know whether naked DNA could ever be transplanted. We were worried some proteins might be involved and essential. So we treated the genome that we isolated from M. mycoides with proteases to remove all the proteins. We added a couple of cassettes to it, one for selection and the other for turning the cells bright blue if this got activated, and worked out ways over a long period of time and very many trials how to transplant this genome into the capricolum cell. We actually have this very sophisticated movie to show you what we think happened. We found a way to get the mycoides genome into the capricolum cell. And for a brief time now, we have a capricolum cell with two sets of genetic software. And so what we think happened is this new DNA started getting read the second it was put in the cell and made a number of proteins. Some of the early proteins that we think it made are restriction enzymes. The restriction enzymes recognize the capricolum genome as foreign DNA and chewed it up. So now we have a capricolum cell. Anybody defining things phenotypically would say this is capricolum. If you're defining it genetically, you would have found in this rare cell the mycoides genome. But what happened was, after a short period of time, we ended up with these bright blue cells. And when we interrogated these cells and sequenced the proteins, there wasn't a single capricolum protein left in these cells. Every molecule in the cell derived from the new genetic code that we put into the cell. So we say life is a DNA software system, and you change that software and you change the species. A lot of people thought this experiment wouldn't work. In fact, feedback we even got after publishing it, people were sure there must be some kind of artifact in this, that we thought the synthetic DNA was obviously the key to proving. But we have a problem where we were now assembling the bacterial genome in yeast cells. So we had to work out a way to get the genome out of yeast to transplant it into the bacteria cells. And so to do this, we developed a new cloning system for cloning entire bacterial genomes in yeast as yeast artificial chromosomes. These are just circular genomes. And unlike what people thought with the human genome, because there was no selection when human yaks were done, simply by selecting these are actually very stable clones. Yeast seems to be very happy to carry on a bacterial genome as an additional chromosome. So we now had the ability, because we had the mycoplasma mycoides genome in yeast, so we could now isolate it from yeast and see if we could transplant it. And we tried it, and it didn't work. And it took us 2.5 years to solve all the problems with this. It turns out methylation was the key to this, and restriction enzymes. In fact, it turns out when we isolated the genome initially from the mycoides cell, it was methylated. And that protected it from the restriction enzymes of capricolum cell. You would have thought with Ham Smith, who discovered these as a key part of this, we might have noticed that sooner. But we isolated all the methylases. If we then now methylated the DNA when we isolated it from yeast, we could transplant it. And to prove this, we removed the restriction enzyme gene from the capricolum genome. In that case, we can take naked DNA out of yeast and transplant it. So this gave us this complete circle where simply by adding a yeast centromere to a bacterial genome, we can now grow it indefinitely in yeast. And people work on E. coli and a few other bacterial species because they can. Most bacteria do not have nice genetic systems. But now we have what David and others have known all along. You have this tremendous genetic repertoire of yeast. So we can make rapid modifications using homologous recombination on a bacterial genome in yeast, isolate it, methylate it if necessary, and transplant it, and just work our way around the circle. And had we had this technology initially, we never would have developed the synthetic DNA approaches. So now, because we had all these and we had the new synthetic capabilities, we decided to go ahead and make synthetically the entire 1.1 million base pairs of the mycoides genome. We did this by starting with 1 kb pieces. We put 10 of those together to make 10 kb pieces. We put 10 of those together to make 100 kb pieces. We put 11 of those into yeast, and yeast assembled the entire genome. We did the transplantation, and it didn't work. So we had to set up what software engineers have of debugging software. We had to develop a biological version to debug the genome to find out why this hadn't worked. We had sequenced this with next generation sequencing, and it looked like it was entirely correct. So what we did is we made corresponding pieces from the native mycoides genome and substituted them one at a time, doing all these transplantations. And we got down to 10 synthetic pieces, and the last piece had to be from the native genome. So we went back and resequenced the piece using Sanger sequencing, and we found a single base pair deletion in the DNA A gene, an essential gene. We corrected that single base pair mutation. 1 out of 1.1 million made the difference between life and no life. So now with the base pair corrected, we did the transplantation, and we got what we're defining as the first synthetic cell. And we're calling it a synthetic cell because everything in it derived from the synthetic DNA. Our biggest concern with this was artifacts and worried about even carrying along a single molecule of native mycoides genome. Even though we had lots of changes to look at, we decided to get to this point, we had to watermark the genome. So in the first genome, we just used the single letter amino acid code to put the names of all the scientists that contributed to the work in there. But that's very limiting. And so, and people also said we were not very imaginative. I was supposed to put in something like 1 small step for bacteria. And so we tried to advance it a little bit with this one. We developed a whole new code for writing the English language with complete punctuation and numbers using the 4 letters of DNA. When we sent this to Science, they sent it out for review. One of the reviewers sent back the entire review written in DNA code, which really upset the editor at Science. And basically, the code is one that's been used for a long time. It was an ASCII code. And we argue this code is much better. We developed one very specifically for biology that puts in very frequent stop codons. Our worry with using the ASCII code, you can end up with very large peptides, and we were worried we might inadvertently create some new toxin just by trying to put a little poetry in the genome. So we also put in a URL, and when we first published this, we didn't tell people what the code said. And as people solved the code in the first watermark, they sent an email to the synthetic genome saying they solved it. And after a certain number of people did that, we published it. So in addition to the 46 names of all the scientists that contributed to this and the institutions, I chose 3 quotations from the literature that I thought would be important. The first is from James Joyce: "To live, to err, to fall, to triumph, to recreate life out of life." That seemed highly appropriate. The second is from American Prometheus, Oppenheimer's biography: "See things not as they are, but as they might be." And the third from Richard Feynman: "What I cannot build, I cannot understand." So we thought this was fine until after this became widely publicized, what was in there. We got a letter from James Joyce, a state attorney, complaining that we had not asked for permission to use this quotation. James Joyce was dead. We didn't know how to do that experiment. But in the U.S., at least, the fair use laws allow you to quote up to a paragraph without any problem. So then we started getting emails from a Caltech scientist saying we had misquoted Richard Feynman. And if you look at this quotation, you'll find this thousands and thousands of times on the internet. And so we argued a little bit with this guy, and to prove his point, he sent a picture of Feynman's blackboard when he wrote the original quotation. And the original one is actually much better. And one of his biographers obviously made a mistake and changed it. So the original quotation is, "What I cannot create, I do not understand." So we've gone back to change the genetic code so that Feynman will rest peacefully, and we'll have this correct version in there. So the chance of having all these things occur in a naturally occurring genome are relatively low. And we think, aside from the fun of this, it's essential with synthetic DNA, and particularly with synthetic genomes, to watermark them in a very significant way, to track them and make sure they don't show up confusing evolutionary biologists as we make more and more complex things. And we're right now, design is very difficult. We have a contest within the Institute and SGI trying to design a minimal genome. And the problem is at least 100 of the genes in the minimal gene set are of unknown function. All we know, if we knock out that gene, the cell dies. So it's kind of hard to do design on first principles when you don't know the first principles of biology. But we are building one, and if one of them leads to life, we should know that soon. But I like to view all these genes that the community's discovered, all the things that come from the Sorcerer expedition and the human gut, as design components. The same way the electronics industry had resistors and capacitors, then ultimately transistors and integrated circuits. Right now we have 60 million. We don't know where discovery is going to tap out. What is the gene complement of our planet? Obviously it's changing dynamically, but it's probably between 1 and 200 million genes. And we have software at Synthetic Genomics for actually designing DNA software. We're trying to build new organisms, so we're putting this to this complete test right now. There's other reasons to do this. Obviously, with the problems we're facing on this planet, we're going to add 1 billion people to the 7 billion we have now in the next 10 years. We don't have sufficient food, water, energy, medicine, housing for the 7 billion. CO2 is increasing a problem more dramatically than I think most people thought it would. So we're trying to recycle the CO2 in a variety of ways. We're using the synthetic DNA technologies already for a new way to make vaccines. This is the meningitis vaccine. It's the first genomic-based vaccine that we developed with Novartis from the meningitis genome, and I think that's coming on the market this year in Europe. But we really concentrated on flu vaccine, because if you look what happened with H1N1, it took about 9 months to get a new vaccine on the market to treat that. If that had been really a pandemic that was expected, it would have been a real problem. My institute's part of this influenza network. We're the major source for sequencing in the U.S. from viral isolates around the world, and we're trying to develop a predictive algorithm to predict how the virus is changing with time. And we're working with Novartis and with BARDA in the U.S. government, and the goal was to get vaccine production changed from 35 days to less than a week. And right now we've perfected this situation. BARDA sends us an email message with a theoretical pandemic viral strain, and we have less than 24 hours to make the new virus. We actually have it down to 10 hours now. We get this to the Novartis scientists who have this multi-billion-dollar facility in North Carolina for growing the virus in MDCK cells. That depends whether adjuvant is used or not. It's certainly enough for massive populations in a very short period of time. So we think we're pandemic ready, and we're trying to get approval from FDA now for the seasonal flu vaccine that we're trying to do predictions well in advance. We're using changed algae cells to try and make proteins, oils, fuels, etc. And right now, omega-3 fatty acids come primarily from crushing fish and extracting it. And many people don't realize the fish oil and the fishy smell is actually algae oil, and it's oxidized when it's in these dead fish. When we isolate it right from the algae without passing it through fish, you can keep it in a much cleaner state. And we have a facility in La Jolla, a large greenhouse there, where we're testing a variety of new strains and ways to grow things. And those of you who might have seen the New York Times article last week, they had a photo of our first major advancement in changing photosynthesis and really changing what we can do genetically with these algae. And we just purchased an 81-acre facility in the Southern California desert for scaling up these kinds of things. To close, people always ask questions about the ethics of creating new species. And we asked these questions before anybody else did. We, early on, before we did the first experiment, asked the University of Pennsylvania to take on a study of synthesizing a minimal genome. And they published this in 1999 in Science, along with one of our series of transposon mutagenesis experiments, and thought we were taking a reasonable approach, and their concern was over abuses of this technology. The Sloan Foundation has funded my institute along with MIT and a Washington think tank to do a series of reports over time on the security risk, and you can download that from our website at jcvi.org. And in 2010, when we announced the synthetic cell, President Obama immediately asked his new bioethics commission to take this on as their #1 charge, which they did, and they published this report in 2010 in December, and you can get this from a number of government websites. And I think this is a very comprehensive report. I think they did a great job. They talk about the potential, but also what's needed is designing a number of new biological tools that could be built into genomes to limit their growth and distribution when we want to. There's probably been tens of millions of experiments that have been done in laboratory E. coli with no known major accidents from doing that with genes from every known species. Why? Because it won't grow outside of the laboratory. In these large facilities, DNA does blow around. If you have a swimming pool, you know you get algae coming from the ocean and the land and other places. So limiting things so they can't survive in the environment is going to be an essential part of this. So I urge you, if you're interested in this space, to look at that. So this is an exciting time as we go from reading DNA to writing DNA to showing that DNA actually contains all the information required for a cell to make everything it needs and to go into a self-replication form. Whether that translates from a simple bacterial cell to a human cell remains to be established, but I don't think that's a huge leap for any of us. Thank you again for this award and the invitation to be here.
Thank you very much. We have time for questions. Please use the mics. Yes, please. Go ahead.
I think you need to stand up and shout. So we do it in multiple ways, but basically because the DNA is so much simpler than human DNA with all the introns and intergenic regions, we just simply do a 6-frame translation of everything as the first pass and do a comparison to everything that's known. But then we go back and do a much more sophisticated gene prediction to whatever extent that will work. So obviously, if it's using the kind of codon usage that's unique to the mycoplasma, that would show up. Whether we would see a totally unique codon bias with that approach, we probably would not. So if it's an alien life form, we probably missed it.
Yes.
Okay, 2 questions. One question.
There were 2 questions.
The oceans. Some locations in the oceans that are more interesting in the sense that they are significantly more diverse population of creatures.
Yeah. So the question is, did we find parts of the ocean that were richer, more diversity? Actually, the stunning thing to us, roughly every 200 miles, about 80% of the sequences were unique. And so instead of the oceans being a giant homogeneous soup, it's millions and millions of microenvironments that are constantly changing. But what we did find that's linked to that, we found over an order of magnitude change in the viral to cell ratio. So in some places we found only 1/10 the amount of viruses that affect the cells, and many people think the populations in the cells are controlled by the viral populations. So that's a huge difference in the viral load and the ability of the cells to go into these huge blooms very quickly. But the diversity has been overwhelming everywhere we've looked.
Second question regarding the killer cell. Did you sequence the killer cell, the immortal cells?
I would ask the next speaker that. I don't know. I assume somebody might have.
There are so many different types of things
Yeah.
that all get called killer cells
Yeah.
that I don't know that it's a meaningful state.
Yes.
Okay, last question.
Yeah.
So when you go to specify the context, do you call yourself a creationist or intelligent designer? And the more serious note, how do you manage public perception about what you're doing? For example, we are living software for designing new things, and we get the reaction of, oh, somebody can just load Ebola and figure it out.
Yeah. Well, what we've tried to do, one of the advantages of this taking much longer for us to accomplish than we thought it would, is we've had basically 15 years to roll out these ideas. We've tried to do it in a very public way. There's been public hearings, public discussion, but it's really stunning despite all the discussion, all the publicity. Each time somebody talks to a different group, it's the first time they've ever heard these concepts. So despite what we think about information transfer in our societies, when it comes to science, it's still remarkably inefficient. So we've tried to be very candid about what we're trying to do, where we're trying to go with it. We've had all these studies on different approaches. And it is powerful technology, and somebody could abuse it tremendously. And it's a very serious issue. So we don't answer it by saying it's not a problem. We answer it by saying it's a very serious problem. But the potential for this in terms of benefiting society, and the Obama Bioethics Commission came up with the same conclusion. The potential gain is far worse than the potential problem. And even it may not be very comforting to people, but the security experts in the government actually worked out there were so many other ways for people that want to do harm to get a hold of anthrax or get hold of the thousands of freezers and labs around the world that have hoof and mouth disease, that they're not going to go all the trouble to synthesize one, but it is getting easier and easier to synthesize them. So we don't want to whitewash it. We don't want to pretend that's not an issue. But what I argue is the same issue with emerging infections, which I think are a much bigger problem for humanity. We need new antivirals, new antibiotics, new vaccines, and new ways to make them very quickly. If we have those, bioterrorism goes away as a serious threat.
Okay, thanks, speaker.